Hello and welcome back. So today's presentation is chapter 8, and of course you can see it's still on chromosomes, but now we're talking about mutations again. And this time we're talking about specific mutations within the chromosomes. We did that in a few chapters ago, where we talked about crossing over with chromosomes, and that's a mutation. And then we saw the equivalent in bacteria. And then we went on to chapter 7, where we saw aberrations in the sex chromosomes. And now we're talking about aberrations in the autosomal chromosomes. So all of these mutations, of course, are aberrations, and but now they're in the autosomal. So chapter 7 and 8 both dealt with chromosomal mutations or chromosomal aberrations. It's just that chapter 7 was on the sex chromosomes, and chapter 8 is on the autosomal chromosomes. So here's our learning objectives for each one here. So a modification at the level of a chromosome. So we can definitely see phenotypic variations that result from changes of the individual genes. And some chromosome mutations and aberrations, you could have that as a total number of chromosomes. So the list right here, the 4 items, are 4 items right here, are they're the 3 or the 4 types of mutations that we'll see. And again, mutations and aberrations really are the same thing. Sometimes you'll see an aberration just with pertaining to just a duplication, but that's not really accurate. They're all pretty much in the same category now of either chromosomal variation, deletion, duplication, or rearrangement. So we'll look at all 4 of those coming up. So here's our variation in chromosome number. So we're going to start with our terminology and origin as well. So so here our variation of what our normal or our wild type is that we have 46 chromosomes, but they're arranged in pairs. So we have 23, a set of 23 chromosomes. Each set or each pair has 2 chromosomes in it. And 22 of those pairs are homologous, meaning they're the same. And one set is not. One set is the sex chromosomes, and of course they're different. So again, 23 sets, or we vary. 23 or 2 haploid sets, 23 total sets. Each set is a haploid set. And but there can be variations in that where the number of chromosomes change. So a chromosomal mutation or an aberration, again, they're both the same, that results in a change in the total number of chromosomes. So you can either have a deletion of a chromosome or a duplication of a chromosome, or a rearrangement of the genetic material within or among all those chromosomes. So that one is so. The first one is pretty clear. The total number of chromosomes changes, and we sort of saw that with the X or the sex chromosomes with Klinefelter and Turner syndrome, where they each got an extra X chromosome. So the total number of chromosomes in both of those varied, and it just happened to be the total number. The number variation happened to be within the X chromosome or within the sex chromosomes. Here we're talking about the same thing. It's just that now in this chapter we're talking about a change in the number of the autosomal chromosomes. And you can either have a deletion of an entire chromosome or a duplication, or you can have a deletion or duplication within a segment. So you can have the whole gene goes, the entire chromosome goes, or some segment gets deleted or duplicated. And then rearrangements of genetic material within or among chromosomes. Again, a lot of that is going to be that crossing over where this piece goes here in exchange for a reciprocal amount here. But we'll see some variations on that as well. So aneuploidy is the term for variations in the number. Typically, it's ploidy. The last part here is really referring to the number ploidy is some variation in chromosomal number. So in aneuploidy, chromosome gains or loses one or more chromosomes, but not the entire set. So you have your entire set, your 46 chromosomes, but you can gain or lose one or more within that set. Monosomy is the loss of a single chromosome in a diploid genome, and trisomy is the gain of a single chromosome in that diploid genome. Trisomy, the common one that you're probably familiar with, is trisomy 21. So trisomy 21 affects chromosome 21, and they get three of those. A person with trisomy 21 has three chromosomes of the number 21 chromosome. The common name for that is Down syndrome. So again, Down syndrome is also trisomy 21, and it's where the person with Down syndrome has three chromosome 21s. And then euploidy is a complete haploid sets of chromosomes are present. So you get a variety. Instead of just one set of the chromosomes, you get multiple sets of the chromosome. And polyploidy is more than two sets of chromosomes that are present, and triploidy is three sets of chromosomes that are present. So most of the time, when there's more than one set of chromosomes, when you have your 23 pairs, when you have duplicates of those, or like more than two sets of chromosomes, oftentimes these are not consistent with life. And I sort of went kind of fast over the Down syndrome part. Obviously, Down syndrome, that's a trisomy. So we know many people with Down syndrome. So a person can be born with Down syndrome, and that's very consistent with life. But there's two other trisomies as well, trisomy 13 and trisomy 18. So trisomy 13 is, if we have trisomy 13 and trisomy 18. So 13 is Patau syndrome, and 18 is Edwards syndrome. And neither of those that were consistent with life. Both of those, and same with monosomies. Monosomies are not consistent with life either. Children or infants or fetuses with Patau or Edwards syndrome or any kind of monosomy, they don't survive to term. They're pretty much they're pretty much would be a stillborn. Actually, I think stillborn. The child will be born dead, deceased. But oftentimes it's a miscarriage, and like the mother does not carry full term, like with a traditional stillborn would be full term, and then the child would die from a non-genetic disease. Like a stillborn, oftentimes the umbilical cord wraps around the neck, and the baby would asphyxiate. So that would be, and it would usually happen like hours before birth, or even weeks. But here with Patau syndrome, Edwards syndrome, and any kind of monosomy, the mother would not carry the child that long before the spontaneous abortion occurs. So here's some variations or a chart that goes over or restates everything. So again, aneuploidy is you have your 2N set, but then you get plus or minus an X chromosome. And then under aneuploidy, monosomy, disomy, trisomy, and tetrasomy all fall under an aneuploidy. And the monosomies again are not consistent with life. Disomy is just your regular set of chromosomes, and then if you get an extra with trisomy, that one we see Down syndrome is compatible with life, but not Patau or Edwards syndrome. And then euploidy, we have our diploidy here, 2N. That's actually the normal the for humans. And then polyploidy, triploidy, and you can see the variations here happening. So some other disorders or non-disjunction, And again, we saw that too, where Turner and Klinefelter syndromes were a failure of disjunction, or a non-disjunction, but again, it was the sex chromosomes. So here we're talking about the same thing just happening in the autosomal chromosomes. So a non-disjunction defined as a failure of one or more pairs of the homologous chromosomes or sister chromatids to separate during cell division. And it usually results in an abnormal distribution of chromosomes to the daughter nucleus. Both chromosomes go to one daughter cell and none go to the other. So you get a chromosomal variation, and the paired homologs again fail to disjoint or fail to segregate, and it happens in meiosis 1 or 2. And oftentimes it's the cause of a spontaneous abortion, and that would be the case with Edwards or Patel syndrome when it's chromosome 13 or 18 that failed to segregate. But 21, trisomy 21, is consistent with life. So here's that chart with cell division, and we can see here what's happening with the disjunction or normal disjunction. So you see here in the first division of non-disjunction, if it happens, we have your chromosome pairs right up here, and one pair should go each, and then each pair separates. But here, the failure to segregate happens right off the bat, and so nothing's coming down this lineage here, and you wind up with trisomy here, and then you get a 1 or a haploid gamete over here, just contributing here. But then when the disjunction happens further down, or in meiosis 2, you get the normal disjunction right here, and you get normal disjunction here, and you see this haploid gamete pairing up, and the haploid gamete goes across the board. But you get the monosomy right here, and that's not consistent with life, where you get the trisomy here. And then if the trisomy is 21, that's okay, that's Down syndrome. But 18 and 13, or Patel and Edwards syndrome, or I said that backwards, 13 is Patel and 18 is Edwards syndrome. And again, neither of those are consistent with life. And monosomy and trisomy result in a variety of phenotypic effects. Monosomy would be a loss of a chromosome, so you produce 2n minus 1, and then although one copy remains, it would be a lethal copy and the organism's not viable. So monosomy unmasks recessive lethals. So if you have a recessive disease, that would be, or a recessive disorder, that's a lethal or deadly disorder. That's often why the organism is not viable or doesn't go full term. And then haploinsufficiency is when one copy is not sufficient for an organism to survive. So oftentimes you hear the term "it's not consistent with life." That would be, I guess, more like the layman terms in telling a mother that her child is, she's not going to carry a child full term. So again, if genetic testing early on, like amniocentesis, reveals a monosomy, haploinsufficiency is the genetic term where one copy is not sufficient for the organism to survive. But the way that usually gets presented to the patient is that the fetus isn't viable or going full term won't be possible. So trisomies again, trisomy is your 2n plus 1 chromosome. So it's the addition of a chromosome, and it produces more viable organisms if it's trisomy 21. Plant trisomies are often very viable, and you see an altered phenotype, and that's what you get here. The trisomy, the leaf is the leaf right here instead of normal. You get this prickly, pointy leaf right there. So you get definite phenotypic changes. Down syndrome, you get phenotypic changes as well. Down syndrome is trisomy of chromosome 21, so you get an extra 21 chromosome. You get 3. And there's 12 to 14 characteristics. On average, affected individuals express 6 to 8 of those characteristics. And here's some, actually, I didn't actually write them down, but I didn't put them on a slide. But actually, basically, what those characteristics are. So here's the karyotype in the gray block right here, and you see this part. You see the three 21s. But then when you look at the phenotype, the person right here, you get the large epicanthal folds right here. Now this lady's tongue it's sort of protruding, but more often than not, the tongue is usually protruding out. Not like the person is sticking out their tongue. It's just a normal resting stage where the tongue protrudes a little bit more. And then also there's intellectual disabilities associated with trisomy 21, varying degrees of disability. Some are very high functioning, others are not. Typically they're short, they're usually heavy set, have a weight issue. They usually have a very round face. Usually it's hard to tell in the lady right here, but typically a child with Down syndrome usually does have a very round face. So the Down syndrome critical region of chromosome 21 is where all the phenotypic aspects are associated with the syndrome. The origin of the extra 21, it's a non-disjunction. They fail to separate during meiosis. So the homologs don't separate or disjoin in anaphase 1 or 2. And it leads to that N+1 gamete. The ovum is the source of about 95% of trisomy cases. So that means the mother is the one that passes this trisomy 21 on to the children. And there's an increased incidence with the woman's age, but there's a lot of controversy around that now. That had been the big understanding. It was that the older the woman is, the more likely she is to have a child with Down syndrome. But then it's getting a little harder to prove that. There's so many other factors that could be influencing the reason for a down child. But if some of the thought now might be it's just it's not the age, but maybe it's a delayed onset of having children. But again, there's still a lot of discussion on that area. So genetic testing is it could be quite extensive. Amniocentesis or chorionic villus sampling, they're the two common ones. Fetal cells are obtained from the amniotic fluid from in an amniocentesis, or the cells are obtained from the chorion of the placenta. So the chorion is the chorionic villus sampling, and both of those tell a lot of information, give you a lot of information about the genetic health of the child. And then there's also the non-invasive prenatal genetic diagnosis. So fetal cells and DNA are actually obtained from maternal circulation. Amniocentesis and chorionic villus sampling are the two most prevalent. I think it's 22 tests that are mandated, but there's hundreds and hundreds of tests that you can actually get. The 21 mandated ones are for like the really common disorders. But pretty much if you have a disease running in your family, you more than likely can get some kind of a genetic test to tell whether or not it's going to be passed to your child. So trisomy 21 is the common, or the one that we're familiar with, the Down syndrome we're familiar with. But familial Down syndrome is more like a family related, like one that runs in the families. And it's a translocation of chromosome 21. So Down syndrome could be inherited or it could be a random kind of thing. So the familial one is where Down syndrome would run in the family. And the translocation of the chromosome, we'll understand when we get to section 8.8. We're going to be talking about translocations then, and we'll see back in that section what that means. And then aneuploidy here again is the trisomy 13 and 18, Edwards and Patau syndrome. Both trisomies Both trisomies in this case, they're not consistent with life, but they can survive to term. In some cases, the mother can carry full term, but the child would manifest severe malformations and would die very early. It's very lethal. So, dying very early means within minutes after birth. If the mother actually has a live birth, the child would be so severely malformed that within the first few minutes of life, the child would pass. And then a lot of characteristics in general with any human aneuploidy is some level of mental retardation, growth failures. So you see a lot of the child would be very short. Low set or deformed ears. The ears would be more often they're very low set, or they just didn't. When it says deformed, they just didn't develop fully. When they don't develop fully, that involves like the ear canal and a variety of the components of the ear itself that don't fully form, and then that's what causes the deafness. Also, heart disorders, the atrial septal defects or ventricular septal defects, either of those in the heart or both and within the heart, deformities of the heart. And then abnormal polymorphonuclear granulocytes, that's an immunology issue where you don't have you don't often make your immune system is basically compromised. You don't make enough macrophages or neutrophils to ward off diseases. Microcephaly is often some kind of a issue with the brain. Oftentimes the child is born with a cleft lip or palate or an extra finger or perhaps deformed fingernails. Kidney cysts are often common in aneuploidy. Double ureters are umbilical hernia, so it's hernia within the umbilical cord, or development of uterine abnormalities that leads. That often means sterility, and then cryptorchidism also is a possible characteristic of an aneuploidy condition. So the trisomies are often found spontaneously in aborted fetuses. Again, if the child actually does is a live birth, minutes after the birth, the child would die. The trisomies account for about 20% of all conceptions. Autosomal monosomies are pretty rare. They're seldom ever found. It suggests monosomic gametes may be functionally impaired and then just don't ever turn into a viable embryo. Polyploidy, in which more than two haploid sets are present, is prevalent in plants, but not really in humans. So polyploidy is more than two multiples of a haploid chromosome that's found. So you have 3N, 4N, or 5N, and that's what this table is showing again. You can see the breakdown. So an origin of polyploidy is the addition of one or more sets of the chromosomes identical to the haploid complement of the same species. So it's not a mixture of species happening within the species. And then the combination of chromosome sets from different species is a consequence of hybridization or allopolyploidy. So autopolyploidy is each identical set of chromosomes is identical to the parent species, and it arrives either through a diploid gamete is produced or two sperm fertilized and one ovum, which is pretty rare. It's more prevalent in natural populations, and but they produce balanced gametes basically is the goal. And then allopolyploidy is hybridization of closely related species, so more common in plants again. A hybrid plant may be sterile and it can't produce the viable gametes, or the chromosomes are not the same and they don't synapse in meiosis. And then endopolyploidy is a condition where only certain cells in the diploid organism are polyploid, and a set of chromosomes replicating repeatedly without a nuclear division. And so that's basically uncontrolled cell growth right there. And you could see that, or you do see that more often, a common occurrence of that in cancer cells. So you see that prolific cell multiplication. So they're replicating repeatedly, but they're not really dividing the nucleus, or you don't see any chromosomes separating or anything, but you end up with this prolific cell growth that gives you a tumor. So you can have variations occur in the composition and arrangement of chromosomes. So that's where you get your deletions, duplications, inversions, non-reciprocal translocations, as well as reciprocal locations. So that's a variation in the composition and the arrangement of the actual chromosome. So prior to this section, we were looking at the chromosomes moving around, like adding a chromosome or deleting. Here we're looking again at the one chromosome, and we're going to see some sections that get deleted or duplicated or inversions occurring. So that's what we're going to explore now. So here's our chromosomal rearrangements. Let me make that a little bigger for you here. So in A, the section D is what gets deleted. So you end up seeing that the chromosomes still join together, but it's just a little shorter. And then a duplication, let's see, it's A, B, C, D, A, B, C. So BCD is what gets duplicated. It's not just the D that gets duplicated. You have your A, B, C, B, C, D. BC is what gets duplicated. So you have your BC here, and you get BC duplicated again, and then you get your D, E, F, and G. So you just get a duplication. You have the original BC, and then it gets duplicated over here. And then the inversion right here is with B and D. So you have your A, B, C, D, but then you get A, D, C, B. So the D and the B, it got inverted or swapped spots. And then down here in non-reciprocal translocation, you wind up with some of the blue chromosome cutting off, where you have your A and B, and the A and the B right there come off and go over this chromosome. So the new chromosome is a lot longer now, but then the old chromosome shorter. And then the reciprocal translocation is that crossover that we were looking at, where you give where the two chromosomes combine, but it would be like HI and J over here. It goes over to it comes off, and the AB comes over here, and then the HI and J goes over in place. There's just this reciprocal in the amount. Notice that it's not a 1-to-1. So you get so you get the AB right here. You get the AB. And then over here you have the HIJ, and then over here you get the AB. So that's the reciprocal change. They notice the centromere stays the same, and so they just swap spots. But you can see that on the orange, the HIJ is bigger than the AB. So it's not it's not a 1-for-1 reciprocal. It's just pieces of the chromosomes are non-sister chromosomes or change, or non-sister and non-homologous are the same. So it's not. It means that the two chromosomes are not the same pair. So it would be like one from chromosome 1 and one from chromosome 2. Or maybe chromosome 1 and chromosome 3, or chromosome 20. So two different chromosomes are going to give each other or exchange information or exchange material. So alterations of chromosomes, deletions and duplications or alterations, and it changes the amount of genetic information on the chromosomes. So you can see that right here. Even if it was a 1-to-1, so here it's not. You have three regions coming off of this one chromosome and 2 coming off the other. So that definitely changes the amount of genetic information on each chromosome. And even if it was a 1-to-1 correspondence, it still is, it still changes the total amount of the genetic information because they're not sister chromatids. Even when they are sister chromatids, they're just rearranging where the alleles show up. But here, they're two different chromosomes. They're not homologous, so they have different genetic information on it. So it completely changes the genetic makeup of each chromosome, because what was originally at locus 5 now has something else at that locus. And then inversions and translocations, genetic material remains the same, but then it just gets rearranged. So that's the one back here where the inversion and duplication, so the genetic material just gets rearranged. It's the same, but it just gets changed around a bit. And then a deletion is a missing region of a chromosome. So deletion, something got taken out. So it's a missing region. So if the chromosome breaks in one or more places, a region can drop out. A portion in between the breaks can be lost for good. A location of a deletion can vary. You can have material deleted off the terminal end, or intercalary deletion is the interior of the chromosome. So it's not the ends of the chromosomes, it's everything between the ends is fair game for deletion. So terminal deletion is at or near the end, where the intercalary deletion is within the body of the chromosome. And then a compensation loop is a buckling out of the chromosome. It forms a little loop, and it's a synapsis between chromosome with large intercalary deletions and a normal homolog. So it requires an unpaired region to loop out of the linear structure, and here's what that looks like. So you get this you get this little looping that occurs right here. You get the looping, and then after it loops, somehow or other something gets cut off there, and the little donut gets lost for good, and then these two pieces stitch back together. You can also have it. You can also see something here where you're forming a deletion loop right here, and we can see right up here where they kind of it loops around, and then you get that part of overlap right there. Well, this one right here doesn't give you the overlap, but these two pieces still will join together and lop that part off, and that loop gets deleted again. So a deletion in the terminal part of chromosome 5 leads to the disease Cry-Duchat syndrome, and it's considered a segmental deletion. And so a little segment of the terminal part of chromosome 5 is actually deleted. So it's not just one nucleotide, it's a little segment that's deleted. And the name comes from the fact that infants often cry very similar to the meowing of a cat. And like a cat's meow that's very loud would be kind of like what the infant's cry would sound like. Infants with Cry-Duchat also have anatomical malformations as well. And here we see the karyotype, and it's chromosome 5 right here, and that end got lopped off. So the only way to know that, so you have to look at the karyotype, and then you have to compare it to a normal, because some in the normal cases, like you can see number 3, one's curved and the other's not curved that much. Same thing with 10, you see that. So you see the normal part, but you would have to take this and look at it, maybe a fish technique or some way of visualizing the chromosomes to see that it's not a matter of just being short, or something really was cut off of that. And a trained cytologist would get that right away, but then would also count the base pairs and do a sequencing to see, just to make sure that it really is a part of chromosome 5 that got cut off. And then the phenotypic changes, you can see in the gentleman's mouth right there, often and the eyes as well. Very small eyes, very much like slits, very indented, but then a much larger mouth with the upper part protruding a little bit more. And so they're the typical characteristics you would see again with someone with cry do chat. And then a duplication is a repeated segment on a chromosome. So the duplication, like we saw with BC in that last example, it just gets repeated in that chromosome. And a single locus is present more than once in the genome, and it can produce, or you could also get a compensation loop. It could arise from unequal crossing over. So in the one example we saw, it was three crossing over or the sharing, the translocation. It was three segments of one and two segments of the other. So that's an unequal crossing over, an unequal exchange of information between synapse or changed chromosomes during meiosis. So here again is these duplications where you see materials exchanged or crossed over between 2 and 3, chromosomes 2 and 3. And so they result in a different in a different length, and but then you can see where you're going to get some of the duplications can occur in this case as well. So those duplications again are right over. Oh, sorry, they're right over here. So you get the duplication of C. Yes, the A, B. So C gets duplicated. And it only happens in this one here. In this one here, you get a duplication here. But then here you get a deletion right here. You've got A, B, D, E, and F. So C is deleted in chromosome 2. But then you get a duplication in chromosome 3. And then 1 and 4 are okay. They didn't change. Then a bar mutation is in the fruit fly, and it's the duplications cause phenotypic variation. So you have the bar-eyed phenotype in the fruit fly results from a duplication, and they have narrow slit-like eyes, and it's an inherited phenotype, or the mutation itself is inherited or passed along. So that's what that looks like. And then gene duplication may play a role in evolution. Gene duplication is hypothesized to be a major source of new genes. So the hypothesis was were supported by discovery of genes with substantial amounts of DNA sequences that were in common, but distinct gene products. So an example here is the gene encoding for trypsin and chymotrypsin have very similar sequences, a lot of sequences in common, but they both give you some kind of a different product. They both give a result in some kind of a different product there. And so that's where you get that it's a major source of new genes, duplication. And that would occur like during transcription and translation where it just encodes for something different. When you get a duplication in there in the chromosome, the translation and transcription and translation is going to give you a different set of amino acids and consequently a different protein change or protein chain. And then inversions rearrange the linear sequence, and that makes sense. So an inversion is a rearrangement of that linear gene sequence. You don't lose any genetic information. Just like a duplication, you don't lose, you gain. But then here with an inversion, it's a rearrangement of the sequence without losing the genetic information. And a segment of the chromosome turned 180 degrees within the chromosome. That's one way of an inversion happening. Another one requires 2 breaks in the chromosome and then the reinsertion of the inverted segment. So if I drew a chromosome here, if I just drew this blue chromosome here, and I'm just going to put the red over here as the segment of interest. So you would get a break here and a break here, and this red segment comes out. And then the red segment, if this is the top and this is the bottom, it gets turned upside down. So now you have the bottom up here and the top here, and then that's what gets inserted back. It gets reinsert the inverted segment. And it could occur from chromosomal looping, where when you have the loop and the loop gets lopped off, so the loop gets cut off, and then you get these two segments joining. They join here. Or what could happen if this gets lopped off before the two arms. So let me draw that a little neater. So that's really kind of where this one looks like. And then if you cut this off, the two strands come together to form one. But while they're this way, while they're migrating together, what could happen with this cut off loop is it looks like it's always going to be a loop, but it doesn't have to. It could change, it could flatten right out, and it could invert. So if this is 1 and this is 2, it could flatten out and change like a 2 to 1 right there, and then reinsert itself in that inversion where you get the second part right at the beginning and the first part at the end. So that's how chromosomal looping can contribute to an inversion. And then you get a few types of inversions. One is paracentric inversion, and it doesn't change the length of the two arms of the chromosome, and it also does not include the centromere. That's not a part of the inverted segment. Pericentric inversion is where the centromere is a part of the inverted segment, so the length of the two arms are going to change when the centromere is included in the inversion. And then the inversion loop, inverted and non-inverted chromosomes and meiosis pair only if they form an inversion loop. So you'll have to get that little loop, and then they rejoin and pair. And then inverted or inversion heterozygotes are organisms with one inverted chromosome and one non-inverted homolog. It could be paracentric or pericentric. If it's a pericentric inversion crossover, one recombinant chromatid is dicentric, meaning it has two centromeres, and the other is acentric, which means it has no centromere at all. And then pericentric inversion crossover is the crossover between pericentric inversion and non-inverted homologs. So this bottom line got covered, but I hope you can see it in the PowerPoints. But what it says is recombinant chromatids have duplications and deletions. So when you do any kind of recombination of chromatids, you can pretty much be assured to have a duplication or a deletion at that point with those. And then our translocations alter the location of the chromosomal segments in the genome. And the reason is that it's a movement of chromosomal segments to a new location. So when you move your chromosomal segments to a new location, it's going to alter the location of those segments within the genome or alter their location. So a reciprocal translocation involves the exchange of segments between two non-homologous chromosomes. So that's pretty much what we were looking at in crossovers. The genetic information can be lost or gained, depends on how reciprocal they are. Like in one case where we saw the 3-to-2 ratio there, or the 3 segments to 2 segments, it's a good chance that some kind of genetic information got lost there. Actually, in the one we saw a duplication, so it might have gained some. And here's the reciprocal translocations. Two possible segregation patterns. One is an alternate segregation pattern at the first meiotic division, and that would have a complete complement of genetic information. But an adjacent segregation leads to gametes containing duplications and other deficiencies. The other deficiencies could be deletions, it could or duplication, more duplications. And if they participate in fertilization in animals, the offspring usually don't survive. So the parent would be semi-sterile. Fully sterile means they don't conceive, but semi-sterile means they would conceive, but not really carry the full term or go full term and end up losing the child. And then a Robertsonian translocation involves a break at the extreme ends of the short arms of two of the non-homologous acrocentric chromosomes, and that means the small segments are lost, and then large submetacentric or metacentric chromosomes end up getting produced, and that would be an example of that familial Down syndrome. The origination of Down syndrome that's inherited is because of a Robertsonian translocation, not the trisomy. So familial Down syndrome is not trisomy. It's often a Robertsonian translocation, and here's what that looks like. So you have chromosome 21 at play here. It's so trisomy 21 is non-familial. The familial or Robertsonian translocation involves chromosome 21 and chromosome 14. So one of the chromosome 21s translocates. It just kind of floats on over to that other or the one next to it. It just kind of just attaches onto the bottom there. So that new chromosome is often called a translocation carrier of chromosomes 14 and 21. And then when you see the formation of the gametes, when you see the formation of the gametes, you see here's the one normal, and then you're pairing up. Here's the ones that segregate, and you get you get the first two, and then you get this one here that's not normal. The translocated carrier 14 and 21, and you see it sitting right there as well. And here's how you get if it went on this route, you'd get the normal. And here's the translocation carrier. Here's trisomy 21, that's regular Down syndrome. And then here's the monosomic, and that's the lethal. The other chromosome's not there. So monosomic is not consistent with life. Well, there's fragile. So we saw a lot of chromosome changes, and so we saw deletions, duplications, translocations, and what's happening in each of those is that the chromosome is breaking. Really, so they're fragile sites in human chromosomes, and those fragile sites are more susceptible to breaking. And consequently, if they're more susceptible to breakage, a consequence of that is the deletions, duplications, translocations, all the things we just looked at, and they're all due to these fragile sites. So, again, a fragile site by definition is that that particular site on the chromosome is susceptible to breakage when cultured in the absence of folic acid or other chromosome or other chemicals. The sites indicate regions of not tightly or non-tightly coiled chromatin. And Fragile X syndrome is an example. It's a folate-sensitive site that's on the X chromosome. And it exhibits FXS, which is the most common form of inherited mental retardation. It's a dominant trait that gets passed on, and it's caused by a trinucleotide repeat in the FMR1 gene. So a trinucleotide repeat, or three of the nucleotides that get repeated, or a nucleotide repeating 3 times, either of those. So in this case here, a trinucleotide repeat, a sequence of 3 nucleotides, is repeated many times, and it expands the size of a gene. The 3 nucleotides, again, it could be a single nucleotide that gets repeated many times. A single nucleotide that shows up in a sequence of 3. So it could be AAA is what I mean here. 3 of the nucleotides, or it could be TCG, any combination of those nucleotides, but it's 3 of them that gets repeated many times, and when it gets repeated many times, that of course expands the size of the gene. And then genetic anticipation is a term associated with the number of trinucleotide repeats that increases with every generation. So as these trinucleotides, the first time it manifests, it'll get passed on to the next generation, and the repeats could increase. And then every time you pass it on, every time that gene gets passed on to the next generation, it keeps increasing. So genetic anticipation is the anticipation of the trinucleotide repeat being increased. Carriers have about a carrier of Fragile X syndrome has 55 to 230 repeats, but so they're not affected. They don't have the syndrome at that point. But when you do, or when you are diagnosed with Fragile X syndrome, it's because you have over 230 trinucleotide repeats. And then Fragile X syndrome, you can see some of the breakage points right here, right in this part here where you're looking at. You can see the twists and turns and changing of the chromosome right there. So there's a link between autosomal fragile sites and lung cancer that have been reported. So the defect associated with the formation of tumors oftentimes is the FHIT gene on a fragile site location, like fragile 3B or fragile site 3B. And that gene, if it's at that location, gets altered or ends up being missing in lung cancer cells. It's a normal protein product of this gene is absent in other types of cancer cells. So the protein product of that gene's not present in other types of cancer cells, altered in lung cancer but not present in the other. And that is everything for this chapter. So we will see you in chapter 9.